This is a brief overview of morphological abnormalities in cattle sperm. In a normal sperm cell, there is a head, a tail, and a midsection. The head of a sperm cell is rounded. In cattle, this head is rounded not quite like an egg. It's flat at the base where it connects to the tail, and to me, it seems a bit like the shape of the end of a Q-tip. The acrosome region is the anterior portion of the head, and it contains the chemicals that are involved in the acrosome reaction, where the zona pellucida of the oocyte wall is broken down so that the nucleus can deposit its genetic material in the oocyte, hopefully fertilizing it. The tail is technically called a flagellum. It is a single strand that protrudes out of the posterior portion of the head, and it flails to propel the cell forward. The mitochondrion is somewhat a piece of the tail. It's technically called the midpiece of the cell, and it produces the energy for the flagellum to flail and produce that movement. A normal sperm cell looks something like this. This visual in the corner is not distinctly a bull sperm cell, but it does have a nice visual of the placement of the acrosome as related to the nucleus, as related to the mitochondrion, and I believed it showed that well and better than a picture of a microscope would. You can tell distinctly in this picture where the mitochondrion midpiece region is, because it appears thicker and darker than the rest of the tail, and these heads do have that very distinct Q-tippy shape. Abnormalities are always going to occur in ejaculates. There are never going to be perfect ejaculates with completely normal sperm cells all across the board. Abnormalities will always be present, but the severity and density of these abnormalities is what determines whether or not it's going to affect fertility. One of the more common abnormalities in cattle sperm is the pyriform abnormality, which causes the head of the sperm cell to become narrower in the nucleus, giving it a pear shape. Other deformities in the head of the sperm cell include misshapen acrosome regions, craters in the nucleus. There are just general abnormalities that don't have a specific name that would fall under the term abnormal contour. There are also commonly issues involving the flagellum. They can be bent and they can be coiled, they can loop around themselves. Sometimes a head will have two flagellum attached to it. Sometimes there will be two heads attached to one flagellum. That specifically is a genetic issue and is not very common. In the case of a free-floating head, it is considered a decapitated sperm cell, and those cells are non-motile because they do not have that flagellum to propel them forward or backward. The Dag's defect is a very interesting defect of the flagellum, and this visual is a representation of the presenting defects that a Dag's defect will cause. Cell D has the flagellum coiled and looped around itself. There is no flagellum strand loose in the substrate, and this cell would not be able to move, I would say, at all. Cell C also has the bending and the looping of the flagellum around itself. However, there is that strand of the flagellum that is out in the substrate, so this cell would be able to move. It just wouldn't be able to move as well as a standard sperm cell, and because of the placement of the end of the tail of the cell, it would move backwards as opposed to forwards, just because of how it would propel itself in its position. Sperm cells can also just be generally underdeveloped. These cells will be small, they won't quite look right, they won't be mature, and they will not be able to fertilize an oocyte. These are just a couple examples of abnormalities in sperm cells. The first image is something that would just fall under the category of abnormal contour. The middle image is something called a proximal droplet abnormality, and this is just when the cytoplasmic droplet doesn't move from the proximal end of the tail to the distal end of the tail, where it should eventually fall off. The third picture is a picture I took through a microscope myself, so it may be a little hard to see, but there's a free-floating head and a bent flagellum that are clearly visible. Then there are a few cells that are a little hard to see, but it would appear that the heads of these cells are very narrow, which can cause problems as far as fertility goes. In addition to narrow heads causing problems, microcephaly and macrocephaly are also morphological anomalies that affect fertility, and those are just when the head of the cell is too big or too small. Morphological abnormalities can occur at any point from spermatocytogenesis through ejaculation. Anything that would occur in the female reproductive tract or during, say, the cryogenic freezing process would not necessarily be the result of a defect in the animal, but it may be trauma or just the environment that the cells are in. During spermatocytogenesis, genetic problems can play a part in just producing inherently incorrect cells. During epididymal maturation, the epididymis may have some type of deficiency or defect that would cause abnormalities or just an outright inhibition of proper maturation in the cell. During epididymal migration is when the proximal droplet moves to the distal end of the tail, and occasionally that doesn't happen. Or during the epididymal migration, issues arise in the flagellum. It may bend, it may break off, it may twist and coil, and that is usually where you'll see the flagellum beginning to express their defects. Sometimes in the tail of the epididymis, when sperm cells are being stored, they can develop abnormalities and defects. The longer that sperm cells sit in storage in the tail of the epididymis, the more likely they are to present abnormalities upon inspection. Additionally, with that, the age of the bull has an effect on the rate of morphological abnormalities, with older bulls usually producing lower quality sperm and not being able to efficiently house those sperm and keep them viable in the tail of the epididymis. Other factors can contribute to that, such as nutrition, just outside conditions, illness, and some drugs may also impact the viability of semen or the viability of sperm after they've been stored in that epididymis. Additionally, the seminal plasma is where it's the environment in which the distal droplet is shed, and occasionally that won't happen, and sometimes it just doesn't happen, and sometimes it is a problem with the seminal plasma itself. And this picture is of a sperm cell with a pinched head. I don't think this is any specific defect. I took this picture myself, but I just thought that was a good representation of an abnormality. I'm not sure if this cell has a tail. It looks like it might have one tucked underneath of it, but I'm not sure. But this would be an abnormality that wouldn't be very common and would still fall under that blanket term of abnormal contour. The effects of these abnormalities are fairly self-explanatory. If there's an issue with the flagellum, the cell will not be able to swim. It just might not be able to move very well. and some defects in the head, in the acrosome region, may make it unable to breach the zona pellucida and fertilize the oocyte. And as a general concept, abnormalities of the morphology of sperm cells results in a loss of fertility or a general reduction of fertility.